Okay, good day, everyone. I'd like to welcome all the attendees and a warm welcome to those joining us from different time zones across the world. My name is Rio Vetter, and I'm the CTO here at NeuroNexus. We're delighted to have you here for this highly anticipated webinar, where we'll delve into the remarkable world of high-density neural recordings and systems, instrumental in helping unravel the mysteries of the hippocampus. Our panel of experts will provide comprehensive insights into this technology and its associated products, followed by an overview of the cutting-edge scientific discoveries stemming from this research. So without further ado, let's jump right into it. I invite you to relax and immerse yourself in the rich content we have in store for you. If you have any questions along the way, please don't hesitate to use the Q&A button located at the bottom of your screen. Our panelists will be on hand to address your queries throughout the webinar. As time permits, questions will also be answered at the end. Our opening speaker, Asiya Gulabchi, assumes the role of principal scientist and product manager at NeuroNexus, boasting, boasting an extensive background in neuroscience. With a PhD in biomedical engineering, specializing in neuroscience and brain technologies from the University of Genoa in collaboration with the Italian Institute of Technology. She brings a wealth of experience to the table. Before a role at NeuroNexus, she devoted 5 years to serving as a postdoctoral fellow in the bioengineering department at the University of Michigan. Today, Asiya will unveil the groundbreaking contributions NeuroNexus is offering to the field. With that, Asiya, the stage is all yours.
Hello, everyone. Thanks, Rio, for your kind introduction, and it's truly a pleasure to be here with all of you today. Let me share my presentation with you all. Okay. I'm anxious, the same as you guys, to witness the findings from Professor Buzaki's team. However, before delving into the new discoveries, I'd like to provide a brief overview of the SiNAPS technology. During this webinar, we will explore how SiNAPS probes stand out in comparison to the similar probes available in the field. Additionally, I will discuss the essential setups, including both hardware and setup required to collect data from these electrodes. For questions, as Rio mentioned, please use Q&A button, and I try to answer as much as the time allows. If your question doesn't get answered, I will reach out individually after the webinar to also set up a one-by-one meeting as needed. One of the most common questions I receive when discussing SiNAPS probe is what's SiNAPS technology? So you may be aware that a significant challenge in the field of system neuroscience is to understand the implementation of complex brain functions at the neural circuit level. To address this challenge, previous researchers have emphasized the necessity for advanced neurotechnologies that are capable of recording with cellular spatial precision and sub-millisecond time accuracy. And the general expectations in the field is that these technologies should be able to monitor both spiking and low-frequency bioelectrical signals in neural network. The ultra-high dense electrodes that made through CMOS technology represents an innovative solution to tackle this challenge. Despite recent achievements in circuit architectures, increasing the number of simultaneous recording channels would lead to the subsequent increase of the chip area. However, thanks to the Corticali team, that they have made this game-changing solution by employing the modular active pixel sensors, which employs the a small front-end circuit underneath of each pixel on the electrode. And this design allows SiNAPS probe to record data from all sides simultaneously. So to provide a comparative perspective, let's consider previous approaches such as like NeuroPixel probes. These probes utilize on-chip circuits for recording from subset of electrodes that are selected from, of course, a dense electrode arrays. So in this architecture, an active switching matrix is integrated under the electrode array area, while the circuit for amplification and filtering are positioned outside of the electrode array area. However, when you look at the SiNAPS technology, the amplification happens directly underneath of each electrode, and that enables SiNAPS probe to achieve the simultaneous recording from all the sides. So now, since that you're aware of the concept, one of the first advantages of SiNAPS probe is the full array readout capability. So on this slide here, you observe the 3D presentation showcasing the 4-shank 1024 SiNAPS probe implanted into the mouse brain. What sets this presentation remarkable is the capability of record and recognize neuronal populations surrounding each individual pixel in real time. This view is made possible through the Radiance Lego 3D model tab, which is equipped with 3 brain atlas models of mouse, rat, and macaque. Another advantage is the suitability of SiNAPS for minimal size packaging. As I explained earlier, the SiNAPS has amplifiers underneath of each pixel with respect to neuropixel. That amplifier is on the base of the device. So that means the SiNAPS base dimension can get lower down that enable us to play with the packaging. So let me give you a better idea of what I'm talking about. So if you look at in this slide, I'm showing 2 available rigid SiNAPS packaging options. One is with 256 electrode, 1 single shank, and another one with 1024 electrodes that distributed across 4 shanks. As you can observe, the layout of electrode sites is organized into 2 columns, and each pixel covering the area of approximately 14 by 14 micrometers square, which the electrode pitch is around 29 micrometers center to center. And in the case of 4-shank probe, the spacing between the shanks is around 560 micrometer. I want to highlight the flexibility of smaller packages, particularly focusing on the dimension of the base that's indicated in these red squares here. So this is really incredible that 1024 channel can be reduced into 50 connector pins. So the pin connector is determining factor that influencing the size of the package here. And this is the area that right now NeuroNexus is innovating and making efforts to reduce the overall size of the package. If you visit the NeuroNexus website and navigate through the product electrode arrays and look at the SiNAPS page, under the references and field data tab, there are published papers by Corticali and IIT researchers available for reading. I highly recommend these resources for in-depth insight into the development of SiNAPS technology. One of the other advantages of the SiNAPS packages that NeuroNexus bring on the table is the opto packages that are specifically designed for optogenetics research. So in this slide, you have a visual representation of both single shank 256 and 4-shank 1024 opto packages. The image on the top right corner showcasing the beta testing conducted in April at Professor Bouzaki's lab. These packages are available today for ordering. One piece of information for the 4-shank probe, we offer the flexibility to assemble up to 4 optical fibers, either directly on the shank or between the shanks. What leverage these opto packages is their customizability. So the placement of the fiber can be tailored to align with your experimental setup. Please reach out to us to discuss the specific details that match your research requirements. In this slide, you're observing an IEEE publication featuring a 4-shank SiNAPS electrode implanted in a virally mediated optogenetic mouse model. Despite the optostimulation being 1 millimeter away from the nearest recording site, the study revealed a fascinating observation that the sites that are further away from the stimulation point also got excited per optostimulation. So this finding is another proof that emphasizes on the importance of having simultaneous recording from all the sites. One of the other advantages is the availability of hybrid packages. So if you're already a NeuroNexus customer, the name hybrid package would be familiar. For those who are not with this term, it refers to the integration of ultra-thin flex polyimide cable with a thickness of 12 micrometer. So again, I want to emphasize that these are different from the circuit flex cables that are available in the market. These are ultra-thin. So currently we have 256 hybrid packages ready for beta testing. Also, we are in active stages of designing 1024 hybrid packages. We encourage you to stay tuned for updates as we continue to advance and enhance the capability of our offerings. Another significant advantage of SiNAPS Probe is the availability of non-human primate package that are designed specifically for targeting deep brain regions. As I discussed earlier, the SiNAPS base can get lowered and minimized. So in this case, it can get lowered to 300 micrometer, and they can get inserted into the cannula structure. So it's following the same concept of NeuroNexus vector array probe. This design allows SiNAPS probe to access deep regions within the primate brain, so making the SiNAPS a more precise option for exploring neural pathways in deep brain regions. So as shown in this illustration, the site layout of 512 SiNAPS probe is organized into 4 columns. So the active length area is around 3.9 millimeter. When considering the insertable lengths, in total around 70 millimeter, accounting for the cannula length, in addition to 6.9 millimeter for the shank length itself. Here is an illustration showing non-human primate user utilizing a chamber on the NHP skull for multi-insertion of the SiNAPS electrode in acute preparation over an extended period. So in this setup, the guide tube serves as a penetration tool to pierce the dura, and the craniotomy can be resealed using this chamber setup for recording sessions and subsequent days. Another advantage of SiNAPS probe over NeuroPixels is the absence of noise coupling issue. SiNAPS signals, they can travel a much longer distance within the system compared to NeuroPixel. We are aware that NeuroPixel amplifiers is larger and positioned further away from the sites, so then resulting in lower noise, and in this case is comparable with SiNAPS noise. However, due to the design of amplification under each pixel, SiNAPS probes do not experience any noise coupling. What does that mean? This means that along the track, as the signal travels from the pixel to the traces, SiNAPS probes are not susceptible to collecting noise artifact like crosstalk noise or antenna noise, while the ground floor noise remains consistent. So this unique feature ensures cleaner and more accurate data collection, enhancing the reliability of experiments and further analysis. Another advantage of SiNAPS probe is the closed loop application capability. So with SiNAPS, we receive a frame of actual data every 50 micrometers without any jittering. This consistent and precise data delivery ensures reliable and accurate closed loop applications, allowing for integration of real-time feedback and control in different experimental setups. Another advantage of using SiNAPS probe is the comprehensive support provided from our team, coupled with the ability to use the full analysis suite in Radiance. So what you see in the image is a screenshot of Radiance Lego platform, a software utilized for controlling the data acquisition system. This software provides a real-time customizable interface, allowing users to optimize their data collection. And the feature, the same probe wireframe that will be used in the experiment. So in this case, the SiNAPS 1024 channel probe is displayed, making the mapping process straightforward. User can easily track down the signal activity of each individual pixel in real time, both graphically and in 3D brain model that I presented at the beginning of this presentation. One of the key advantages of Lego is its ability to simplify the data visualization process. The format that this software saves data is a simple binary format and can be easily converted into other formats for the user that prefer to employ their own offline analysis tools for further analysis. Presented here is the required setup for data collection using SiNAPS electrode. The SmartBox Pro is one piece of the hardware that's needed and function also as a standalone data acquisition system that support up to 1024 recording channel and a sampling rate up to 30K samples per second. It has total of 8 GPIOs in the back of the SmartBox Pro for those of people that are interested in synchronizing the EFS data with other data sources. The SmartBox Pro features 4 ports and each accommodating up to 256 channel recording. Specifically for Synaps integration, only port A is active for interfacing with the Synaps box. This image displays the Synaps interface box specifically designed for collecting data from Synaps probe using the Synaps cable. Currently, the length of the cable is in 2 sizes of 18 and 39 inches. Last but not least, the layout will soon be available. Multiple layouts will be soon available for ordering. Here is an example of the Synaps 8-shank 1024 probe that we beta tested in April and gathered preliminary data with this specific probe layout. The presentations that later the Professor Bouzakis' team, Missy and Anna are going to present will focus on the insight derived from this data collection. As I mentioned earlier, we have provided references on our Synaps page for those interested in delving deeper into the technology. And additionally, I encourage all of you guys to visit our YouTube channel for the latest Synaps videos. So I want to thank my colleague teammates at NeuroNexus and also at Corticali for bringing this innovation to you guys. And also thank you guys for your time and attention to my presentation. So I'm available if you have any question.
Thank you, Asiya, for this insightful overview of the high channel count probe and system solutions. Now let's delve into the scientific aspect. We're thrilled to have 2 accomplished researchers who will present their captivating work. Our first speaker is Mihai Boroslakos. Mihai received his medical degree and PhD in neuroscience from the University of Szeged in Hungary. As part of his PhD work, he developed a multi-channel stimulating and a 256-channel recording system to measure the resulting voltage changes in human cadavers who had non-invasive brain stimulation applied. In 2016, Mihai moved to the University of Michigan and worked in Professor Yusik Yoon's laboratory. He set up an electrophysiological laboratory in the electrical engineering department and validated several recording stimulation devices for in vivo optogenetics. He also contributed to the development of flexible electrodes. In 2018, he joined Professor Yuri Bouzakis' laboratory at New York University. His current research combines brain stimulation with large-scale electrophysiology in behaving rats and mice to study how non-invasive brain stimulation interacts with network oscillations leading to altered behavior. Next up, Anna Masalarova will present her research. Anna earned her MD PhD from Charité Berlin in 2014, working on mechanisms of drug resistance and homeostatic plasticity in epilepsy, lab of Uwe Heinemann. After that, she trained as a neurosurgeon and completed her neurosurgery residency at the University Hospital Erlangen in Germany, 2022. Currently, she is a postdoctoral researcher at the Bouzakis Lab at NYU Neuroscience Institute. She is interested in how epilepsy and neurodegeneration affect hippocampal oscillations related to learning and memory, as well as in neuromodulation for treating memory deficits. At this point, Mihai and Anna, the stage is all yours.
Hey, thank you, Rio. Okay, thank you, Rio, for this really nice introduction. And thank you for allowing us to be part of the testing and development of the 1024 channel SiNAPS probe. So imagine that you are working on your research project and one day a team of hardware and software engineers bring you their most recent technology for beta testing. And this is what happened to us this May. So during this period, we tested multiple different devices, single shank 256, 4-shank 1024, and 8-shank 1024 channel probes. Today, Anna and I are going to talk about data that we collected with the 8-shank devices. So as the name suggests, this probe has 8 shanks. Each shank has 128 recording sites. Each recording site is separated by 28 micrometer in a poly-2 configuration. And here you can see the channel map layout and an actual image of the probe. And the shanks are separated by 280 micrometer, so the probe can record from a 2 by 1.8 millimeter area inside the brain. And the width of the shanks is 80 micrometer, and the thickness is 50 micrometer. So in comparison, the NeuroPixels 2.0 probes have 70 micrometer width and 25 micrometer thickness, while a Buzsáky 32 probe, for instance, has 50 micrometer width and 15 micrometer thickness. So given these differences in dimension, first we wanted to get a sense of the single unit quality that we can record using SiNAPS probes. So for our initial tests, we implanted 3D printed plastic base plates in 4 mice. You can see the base plates used for head fixation in green, and then you can see the probe in scale on top of the animal's head. But on our first attempt to record, we immediately ran into some issues because the current version of the probe allowed limited penetration into the brain due to physical constraints. So we had to remove some part of this plastic base plate, but after we had enough space, we were able to reach our target depth. And another unexpected obstacle was the ground. So recommended by the engineers, first we implanted 100 micrometer platinum wires inside or above the cerebellum, but we also tested stainless steel ground screws. And as you can see, with the different ground options, we were able to record high quality data. Here, each column represented by different colors shows the different shanks. So, and as you can see, we had in this particular session 6 non-functional channels. So we confirmed that both methods can work for ground, but in some sessions, we had hundreds of non-functional channels. So as you can see here in these chunks of data across multiple shanks, we had non-functional channels. And what we found is that when we had a poor CSF ground connection due to a short screw implanted above the cerebellum, we had this issue. So what we did to resolve this issue, we implanted in the same mouse a longer screw. So then we had a really nice, good CSF and ground connection. And on average, we had only 6 to 10 non-functional channels throughout our recordings. So once we resolved all technical challenges, we recorded from the hippocampus, as you can see here in this schematic. And given the 8-shank 2 by 1.8 millimeter coverage of the SiNAPS probes, we can record simultaneously for all regions of the hippocampus. And as you can see, each shank yielded high quality wideband LFP signal. So you can see here an example ripple, and in the zoomed in channels, you can see really nice bursting activity for an example single neurons. So after spike sorting with KiloSort 2.5, we can readily identify the cellular layers of hippocampus, and we can also observe some single unit activity in cortex. And based on waveform metrics, we were able to categorize the recorded single units into putative pyramidal cell types represented by red triangles, and into putative interneurons represented by the blue and the cyan circles. So in this example session, we recorded 225 neurons on 171 channels. Again, here in the background, you can see in gray squares the location of the recording electrodes, and in red triangles, the putative pyramidal cells, and in circles, the putative interneurons. But given the dimensions of the probe, we also wanted to check the amplitude of the signal. So on average, we recorded in this session 85 microvolt single units, and then here you can see all the recorded single units in scale relative to each other. And but as you can see, we recorded quote unquote only single units on 171 channels. So to further increase our chances to record more single units, we implanted the 8 shank probes into the midline cortical areas shown here in this schematic to record from motor, prelimbic, and cingulate cortices. And in this particular session, we recorded more than 400 neurons on 272 channels. And so in summary, for our recording and analysis pipeline included inserting the probe into the brain region and reach our target depth within 30 minutes. Then we waited 20 minutes for the brain to stabilize around the probe. And then we typically recorded 2 hours. And I want to highlight that with 1,024 channels sampled at 20 kilo sample per second, you will record 260 gigabytes raw data per hour. So, but after the data was collected, we converted into kilosort format, and we ran automatic spike sorting that we manually inspected using Cell Explorer. And then in our initial tests, on average, we recorded 240 hippocampal neurons per session. But since May, we collected 27 sessions using 2 devices. And I would like to highlight that one trick to extend the lifetime of the probe is to clean right after the recording in 1% tergazyme. So what we typically did, recommended by the engineering team, is 1 hour soaking in tergazyme, followed by 1 hour soaking in DI water. And in the remaining part of the webinar, Anna is going to talk about how we can use this unprecedented density and number of channels to tackle translational neuroscience questions.
Thank you, Mishi, and thank you, Rio, for the introduction and also for the possibility to record with this new probe. So my part of the talk is going to be about how we assign hippocampal regions to the 1,024 channels. And after that, I will show you how we can look at activity propagating throughout the hippocampus. In my case, these are interictal epilepticon discharges. And finally, how we can use this rich data set to help tackle a problem with human depth electrodes. So Mishi already told you about the unit yield of this probe, and the other big advantage of this high density and widespan probe is the density of the local field potential. And personally, I find the local field potential very fascinating because of the richness of the signal. So basically, if we know how to interpret the signal, even one single trace could be telling us a story. And maybe the most famous story, at least in the hippocampus, is sharp wave ripple, which has evolved from a simple local field potential trace to a concept about organized neuronal activity that is linked to memory formation and memory consolidation, and also retrieval. And to the trained, to neuroscientists trained at looking at hippocampal signals, this ripple trace will tell us that we are in the CA1 layer of the hippocampus. And we can also go further and identify sublayers of the hippocampus, because we know that the CA1 ripple is initiated by input coming from the shaft collaterals to the dendrites of the CA1 neurons. And because of the inward ion flow into the dendrites, we will have a negative local field potential in the stratum radiatum, which will produce a negative wave superimposed by the ripples. So now we have the privilege of looking at not one trace, but looking at basically a whole 2-dimensional picture of the hippocampus. And we can easily see the whole corner monis delineated in this picture. So we barely need analysis even for this delineation. But if we do a current source density analysis of fields, we can even differentiate between the dendritic layer stratum radiatum with the current sinks shown here in blue, and the pyramidal layer with the current sources in red in this case. Here I have highlighted the CA1 pyramidal layer, and for the rest of the hippocampus, we can use the similar principle. For example, for the dentate gyrus, we are making use of the fact that the input to the dentate gyrus molecular layer from the entorhinal cortex, if it's synchronized, it will create a big negative deflection, whereas in the hilus we will have a positive wave. And this is an event that we know as the dentate spike, which is even better seen when we look at the sources and the sinks of the dentate spikes. And now we have the dentate gyrus, and to complete the picture, we can also make use of the single neurons that we identified, because we know that the single neurons, the spikes will be identified on pyramidal layers. So we end up with a channel map for 1024 channels of the hippocampus. And next, I'm going to show you how we can use this to study the propagation of neuronal activity that spreads throughout the hippocampus, and also the neuronal modulation of this activity. I am interested in memory impairment and in oscillations that impair memory in the hippocampus. And one such oscillation is epileptiform discharges. Interictal epileptiform discharges are transient abnormal events that can occur basically anywhere in the epileptic brain. And they are called interictal because usually they're generated in between seizures, so they are not seizures, they are much shorter, and the patients normally wouldn't notice them. But we know that actually from a long line of literature, human and rodent, we know that when an interictal discharge is happening, the patient will have a transient cognitive impairment, and the type of the impairment will be different depending on the region. So in the hippocampus, we could have a naming or a naming problem or a memory problem very short. So what is another interesting aspect of interictal discharges is that we now know that they not only occur in epilepsy, but also in Alzheimer's disease, and not necessarily in between seizures, but they can occur independently of seizure activity. And there are several animal models that express interictal discharges. In this case, this is an example of the CA1 of an APPS1 mouse, a mouse that carries two mutations that promote the development of amyloid plaques. But there are even studies, reports showing that interictal discharges can be seen isolated in the hippocampus of patients with Alzheimer's disease. So we analyzed in the lab, we analyzed the interictal discharges in this specific mouse model in the CA1 region. And we observed that when IDs will happen in the CA1 region, they will interfere with other activities such as theta, and they will also decrease gamma and high frequency oscillations, power spectrum, but they will also suppress ripples for prolonged periods, several seconds. And more interestingly, this correlated with the performance of the animals on a maze. So from these results, it is kind of intriguing to find a way to block interictal discharges, because this could be a potential target for improvement of memory deficits. However, these events are very brief and they seem very generalized. So we need to know in more detail where exactly they are generated and where they originate from, so that we can target possibly the region of origin. And to study this interictal discharges, the SiNAPS probe seemed to be just perfect, because it could show us the picture of the whole hippocampus in 2D. And as you can see here, indeed we see the interictal discharges not only in the CA1, but also, as has been shown from other groups, in the dentate gyrus and basically all over the hippocampal formation. And interestingly, the interictal discharges, they propagate through the layers. So the layers are preserved in this case, as we can see in the change of the phases of the wave. So they seem to they seem to adapt the anatomical structure of the hippocampus to propagate. And to investigate this in more details, we can also look at the current fields of the signal. On the right side, as you remember, this is the current source density profile of sharp wave ripple with prominent sinks in stratum radiatum and sources in the pyramidal layer. We also see similar sinks and sources in the CA1 for interictal discharges, but we see sinks and sources in the dentate gyrus, always along the anatomical structures. I have to mention here, though, that the range, the magnitude of the CSD is very different. So even though this is for CA1, the profile has the same polarity, the magnitude of the interictal discharge is much larger. And I'm going to show this to you now on a better time resolution scale of 1 millisecond. So we will be, if the video works, we will be looking at how this particular event, this is one trace from CA1, propagates throughout the hippocampal formation. I hope this works. So in this particular case, we see that the current sink is starting somewhere around the CA3 region, and this propagates to the whole CA1, as it would be the case also for ripples. But when this plays again, you could observe the dentate gyrus. So now it's coming again. There are also sinks emerging around the dentate gyrus. And this is just one single event. If you look at the time scale, one frame is 1 millisecond, and 0 is the peak in the CA1 event. And we are currently looking at more such events, and there seems to be some heterogeneity. So maybe they don't all start in this way. Maybe they propagate or generate in different regions. This is something I'm trying to figure out now. And hopefully soon we will know more. So if you're interested in this, you can come to my SFM poster and we can discuss. Another thing another question we can answer with this probe is how IIDs modulate neuronal activity in different regions of the hippocampus, as has been previously done for sharp wave ripples. And we know now that neuronal activity in the CA2 region during sharp wave ripples will start a little bit earlier than in CA1. So these are the histograms of firing rates. And we can do similar analysis for the interictal discharges. We can separate the cells in pyramidal and interneurons, as Michi already showed you, and also tag cells who belong to a different hippocampal subregion with the channel map showed you. And this is an example from one session. Let's look at the ripple first. So basically, as we know from many studies during the sharp wave ripple, the firing rate, these are heat maps of individual neurons in the different regions. So the firing rate of CA1 pyramidal neurons and CA2 pyramidal neurons is highly synchronously increased and then decreased, and the same is true for interneurons. But when we look at the interictal discharges, what we see, like the most obvious effect in this case, is suppression of neuronal firing rate after the interictal discharge happens. And this prolonged suppression may explain why other or physiological hippocampal signals are blocked when the interictal discharge emerges. When we look at the firing rate histograms, this suppression is even more obvious, and this is throughout the hippocampus. It's not just in one region, but in the whole hippocampal formation. So last, I think I have time to talk about my favorite problem. And because I'm mainly interested in what happens in the human hippocampus, I would like to share how we study the human hippocampus. So currently, basically the only way to record from human hippocampus is depth electrodes that we implant with patients with epilepsy to look for the seizure focus. And luckily, now electrodes have been designed that contain not only the clinical macro contacts, but they also contain a microwire bundle at the top that allows us looking at the LFP signal and also looking for neurons. And one such standard electrode that we have used in Erlangen is the Benke-Fried electrode. This has, on the top of the microelectrode, it has 8 microwires in a bundle. But this bundle can spread basically randomly. So all these microwires could be close together or they can spread out. And because we cut the electrode at 5 millimeters, I'm just demonstrating here how far away they could be from each other. And we can even have a wire that is outside. So we end up with a recording like this, and we're basically in the dark. We don't know where these channels are, and you can imagine how frustrating it is if we want to study sharp wave ripples with this recording. So one way to hopefully make this easier is at least to know where our signal is coming from. And now we know, I showed you before that, so in these patients we have a lot of interictal discharges. So at least we have an event that we can easily detect and that is kind of conserved. And we also have interictal discharges in the mouse. And even though, of course, these are very different species, I showed you before that the interictal discharges propagate through the anatomical layers. And this is very important because the anatomical layers of the hippocampus are similar in human and mouse. So we also have another advantage now with this probe is that we basically have the morphology of this waveform in any possible place with a high density in the whole hippocampal formation of the mouse. So in a collaboration now with the neuroinformatics lab of Erdem Varol and Tian Xiao, who is a PhD student in the lab, we are trying to figure out a way to compare the human and the mouse waveforms and come up with a classifier that can predict the location of the human electrodes. And I'm really excited about this project. So to sum up, Misha showed you that we collected altogether 27 sessions from 9 animals using just 2 probes, and that high density local field potential and spikes can enable confident region localization on the example of the hippocampus with almost imaging resolution and with such confidence that we wouldn't even need histology, even though we're planning to do some histology to prove this. And high density LFP and spike recording allows mapping out the generation and propagation of both physiological and pathological activities. And lastly, I showed you that these data sets could also help for better analysis of human data. And we would like to thank you again and thank the Tracky Lab and also the engineers who developed this probe.
Yes, and just as a final sentence, I would also like to thank for all the engineers who developed this device, and especially for the software engineering team, because at the initial tests, we needed a really expensive supercomputer to make sense of 1,000 channel data, which it's not extremely straightforward how to do. But over many iterations, right now, we can just even use a laptop or a regular desktop PC to collect reliably the 1,000 channel data, stream it. And it was a really great pleasure to work with you guys to develop this. And we are looking forward to develop this further. And thank you everyone also in the audience for your attention.
Very good, very good. Thank you, Mia and Anna, for that wonderful presentation. We do have a few minutes for questions, if there are any questions from the attendees. While people are thinking about their questions, I do have a question. So based on your research that you guys have been doing up to this point, you know, what new capabilities does a probe like this provide, you know, to extend your research? Is it a function of the additional coverage, the closely spaced electrodes? Yeah, just like to hear your thoughts at a high level on that topic.
So it's actually both, because currently we have like 400 channel recording devices. There are flexible technologies available recording 1,000 channels. But what is really nice with this current 4 or 8 shank version probes, that it's a well-defined anatomy. So then we can go after what Anna just showed, the anatomical structure, because we know at 28 micron resolution where the recording channels are located relative to each other. And on top of that, on average, again, we can collect 2 to, let's say, 300 single units. And just to also address one question in the chat. So we run the kilosort on the entire session, and with kilosort 2.5. If you were to use kilosort 1, most likely you would need to use 1 shank at a time. But kilosort 2.5 was optimized for the 4 shank neuropixels probes, so it can be readily used for the 4 or 8 shank 1,000 channel SiNAPS probes. But so in sum up, I feel that this is also something that we now don't necessarily need to worry about, like where we want to record, because we can always just select a subset of channels that is in our region of interest. And compared to other technologies, there is no limitation, because we are simultaneously recording the 1,000 channels.
Okay, great. Well, I don't see any additional questions. So at this point, I think we can close. I'd like to again express my gratitude to the speakers for their very interesting presentations. I'd also like to acknowledge and thank Corticali team for their support throughout the commercialization process. You know, technology development such as this is not easy, and it takes a motivated and dedicated team to pull this off. So we appreciate everyone's participation in this webinar. The link to the webinar will be available. If anybody is interested, please send us a request. And as we conclude, remember the power of technology to shape a better future. Keep exploring, innovating, and pushing boundaries. Until our next meeting, stay inspired, stay curious, and keep innovating. Farewell and best wishes, everyone.